Good morning, and thank you for joining us. My name is Dawn Cullum, and I am the Senior Events and Training Coordinator for Certara University. As regulators and sponsors, Certara recognizes the growing importance of model-informed drug development. We are committed to educating the next generation of experts in this field by partnering with eminent scientists at designated academic centers of excellence around the world. This year, our Express webinars are being conducted by our partners in Centers of Excellence. Today, Dr. Leonard Kagan is our presenter. After attaining a PhD from Hebrew University, Dr. Kagan joined the Center for Protein Therapeutics at SUNY Buffalo's Department of Pharmaceutical Sciences as a postdoctoral fellow. He continued his research in the department as a research assistant professor before joining the Department of Pharmaceutics, Ernest Mario School of Pharmacy at Rutgers University. He is currently an associate professor and a director of the Center of Excellence for Pharmaceutical Translational Research and Education. Dr. Kagan is a co-author of more than 60 scientific publications. His research interests are focused on the investigation and interactions between biological systems and therapeutic agents and optimization of pharmacotherapy. We ask that if you have any questions, please put them in the question section, and we will get to as many of them as time permits at the end of the presentation. Dr. Kagan, I turn this over to you.
Thank you, Dawn, very much for this very nice introduction. I'm happy to be here. It was great working with Certara, and establishing this partnership was great for our educational program, both for graduate students and undergrad students, and we including some of these Phoenix Certara workshops as part of our curriculum for PharmD students as well. Now, I'm in Department of Pharmaceutics, and as you mentioned, I'm also with some partners from clinical departments. A few years ago, we established a Center of Excellence for Pharmaceutical Translational Research and Education to bridge this gap between basic scientists and bench scientists and really clinical pharmacy in various hospitals in New Jersey. And again, this partnering with Certara was great for us because we can introduce various workshops and use the software for a variety of projects. So today, I'm going to talk about this very interesting modeling project that I participated in. We'll be talking about pharmacokinetics of Midazolam and its metabolites in critically ill pediatric population. Doing studies in pediatric population is very challenging, and as you will see in this presentation, especially in this specific project, subjects that were enrolled in critical condition. So before we begin, I want to acknowledge other participants in the study. The study itself, the enrollment and everything was handled by Athena Zupa at Children's Hospital of Philadelphia. She was a PI of the project, and Gideon Steed was part of her crew. Now, on our side at Rutgers, my grad student, CGU, helped a lot with development of the model and data handling. Now, to provide some background on this project, so majority of pediatric patients in intensive care unit, PICU, can be named. This patient, many of them receive mechanical ventilation, and for that they also need a sedation. And they receive it as a part of the cocktail of various drugs. But in this specific project, we're focusing on Midazolam, which is a short-acting benzodiazepine, and it's widely used both in pediatric and in adult population. This specific cohort that we present today, this was HIP, hypothermia impact of pharmacology. This, in fact, was a part of the larger study sponsored by the NIH, where they looked at therapeutic hypothermia in pediatric cardiac arrest patients. So this was a randomized study, and I'm not going to discuss the larger one, but specifically in this cohort, we looked at pediatric population, and one of the challenges that we faced in analyzing the data was that basically, although we say in children, the population was very different. We had only 39 subjects, but the ages ranged from basically 2 days after birth up to 18 years. And this is a definition of the pediatric patient in this study. All subjects experienced cardiac arrest at some point and were brought to the hospital, and they received the normothermic condition of therapeutic hypothermia, which is defined as slowly cooling down the patient to the target temperature of 33 degrees Celsius. And again, for us, it was an observational study, so there was no specific intervention. They received these treatments, for example, hypothermia, as a part of their regular therapy. And again, we focused on those patients that received Midazolam as part of their regular clinical care. Now, these patients were in a very severe state and critically ill. Many of them, during duration of their clinical care and the study, also received ECMO, which is extracorporeal membrane oxygenation, which is a prolonged form of cardiopulmonary bypass. In pediatrics and neonates, ECMO is often used for a variety of conditions, serious conditions, including sepsis, pulmonary disease, and in this case, it was a cardiac arrest. And there are several very good papers summarizing the potential effects of ECMO on pharmacokinetics, and I have reference down on this slide. But to summarize the major points that might potentially affect pharmacokinetics, and it's something that we evaluated in this study as well, is potential increase in volume of distribution, because the patient is connected to external circuit and you need blood to fill up all these tubes that are outside of patient body. So you're basically adding physical volume, exogenous blood that is added to the circulation. Additional things that may happen due to severity of underlying disease, you can have an increase in extracellular fluid volume. There are various considerations regarding drug elimination. So certain factor may potentially decrease elimination of drugs. So for example, up to 25% of pediatric patients on ECMO have significant renal complication, which may decrease elimination. On the other hand, there can be loss of the drug due to adhesion to these components, like the tubing of the external circulation, and also when the blood is changed, and these, again, circuits are changed, there's a, basically, if there is a drug in that volume of blood, there can be elimination of the drug through these processes. Now, some background information on midazolam itself. It's, as I said, short-acting benzodiazepine. It's very fast onset of action after intravenous dose. It can be given through different routes, and in this study, it was given intravenously, it's a combination of bolus and infusions. It's relatively widely distributed, and volume reported in infants and children was from 1 to 2 liters per kilogram, and even larger in adults. The drug is metabolized extensively through CYP3A4. Most of it goes to an active 1-hydroxy metabolite, and I'll talk about it a little bit more. And protein binding is relatively high, 97%, primarily to albumin. Excretion of the drug happens through the urine with a little bit of unchanged drug, but it's mostly the hydroxy metabolites and glucuronide metabolites. So first the drug undergoes oxidation and then glucuronidation to hydroxyglucuronides. And 1-hydroxy tablet and 1-hydroxyglucuronide are pharmacologically active but less potent than parent midazolam. So now if we move on describing the population basically and data in the study, what we had 39 subjects, 25 male and 14 females, and distribution of ages and shown on the right-hand side. So we had basically very wide range of ages, with approximately 2/3 of subjects at less than maybe 5 years old, and then few subjects that were going up to age of 18 years old. And this was one of the major challenges in modeling the data. Now, 19 patients out of 39 experienced ECMO at some point in their therapy. So it could have been that patients were enrolled while already on ECMO. It could have been that they started the study and they started sampling while they were not on ECMO, and then they came on ECMO and then came off of it. And same happened for hypothermia. 29 subjects experienced therapeutic hypothermia at some point in the study, and again, it might be that they started on hypothermia and continued for the duration of the sample collection. It could have been that they started as normothermic and then temperature was decreased and then increased. So we had a variety of conditions, and again, that potentially contributed to variability in the data that we observed. Midazolam was dosed as a combination of infusion and boluses, potentially based on patient state. So as physicians seen and they wanted to increase or decrease or state of sedation, they administered more or less of midazolam. The observation that we used for the study were midazolam concentration and concentration of 4 metabolites that I'm going to show in the next slide, and we had plasma concentration and urinary excretion data. So as I mentioned, one of the major points that we needed to consider in our study is maturation of the elimination pathways. We all know that in neonates, various elimination pathways, including oxidation, glucuronidation, and renal elimination, are not fully developed, and it take a couple of years until they really mature to adult state, we can say. And as, for example, shown on the right-hand side, a combination of allometry and equations for maturation of elimination pathway usually can better describe the variability in patient clearance. And this is a nice example on the right-hand side from a previous publication, Holford's group. So in our study, we needed to add these equations from here for the maturation of elimination pathway, and we added it on 3 different elimination pathway for CYP-mediated oxidation, glucuronidation, and renal elimination. The equation is shown here, where you have postmenstrual age, basically a Hill function, and you have time to 50% of maturation, and you have a Hill exponent to describe the shape of this curve. Now, since we didn't really have sufficient number of patients to evaluate these parameters ourselves, we fixed those to previously published values for CYP-mediated elimination and glucuronidation. These parameters were estimated in the previous study by Atina Zupa, and numbers are shown here, and parameters for renal elimination and maturation of that pathway were taken from a different publication by Rodin et al. Now let's look at our compounds. So we had data for midazolam, 1-hydroxymidazolam, 4-hydroxy, and their respective glucuronides. So this was the major metabolite, 1-hydroxymidazolam, and it accounts for 60, 70, and some data shown up to 80% of metabolites for midazolam. 4-hydroxymidazolam in previously published studies and FDA data say it's account approximately 3% based on mass balance. So everything that is have this green thing around it, these things that were measured. So we had midazolam itself, hydroxymidazolam, 4-hydroxymidazolam, and then the respective glucuronides, 1-hydroxymidazolam glucuronides and 4-hydroxymidazolam glucuronide. Now, as I mentioned, Dr. Zupa had previously worked on the same compound before, and here's one of the previously developed models. This model was developed on a larger population, but at that time they didn't have the urine data. but this was the base for our model where midazolam itself represented by two distribution compartment, and then you have clearance processes to hydroxy midazolam, 1-hydroxy and 4-hydroxy, and then these are subsequently transferred to respective glucuronides. But as in this study, we had more measurements. This was our model. Again, two compartments for midazolam, and then we have conversion to 1-hydroxy, 1-OH, and 4-OH. And then we have conversion from that metabolite to the respective glucuronide. And as in our urine data, we also observed all 5 compounds. So each compound is also directly excreted into urine. This was our model schematic. So moving forward, the model was done in Phoenix and LME. We used the FOC algorithm for fitting the data. This shows our basic equations. So again, the first equation for central compartment of midazolam, we have 3 clearance processes: direct excretion of midazolam into urine, renal clearance, conversion into 1-hydroxy and 4-hydroxy midazolam metabolites, and distribution to a peripheral compartment here. Second equation describes this peripheral compartment, and then we have equation for each respective metabolite. So the indices here for OH can be expanded as 1-hydroxy and 4-hydroxy. The equations will be exactly the same. There is a production from midazolam, there is elimination towards respective glucuronide, and there is direct renal excretion. And then the last equation, when we're talking about the glucuronide, there is production from respective hydroxy metabolite and excretion into urine. So this was our base equation for this model. Now, as I said, as we didn't have many patients, we had to fix certain relationship based on previously published data. So the volumes for midazolam, the central volume and the peripheral volume, were expressed as a allometric function based on patient body weight. The volume for metabolites were expressed as a fraction of the volume, total volume of distribution for midazolam, and these fraction values were again fixed to previous publication by Dr. Zupa. Now, when we talk about clearance, we had 3 different clearance mechanisms, right? So first, it was oxidation of midazolam into respective hydroxy metabolites, and this is what's shown here. So the typical value for metabolic clearance, total metabolic clearance, this was our estimated parameter, and then it was related to the patient's body weight using an allometric expression, and for each of these patients, the maturation status of the respective pathway was calculated. Now, this total metabolic clearance was split into 1-hydroxy and 4-hydroxy based on the previously published data. Again, 80% goes to 1-hydroxy and 3% goes to 4-hydroxy metabolite. There are some other minor metabolites that we did not include in the model, so these were basically not included in the schematic or the equations. Now, when we're talking about glucuronidation, again, transfer from hydroxy metabolite to glucuronide metabolite was described as shown here. Again, the glucuronidation clearance was a function of allometric function of body weight and maturation of the pathway, and then we had estimated one of these as is. So the transfer from 1OH to 1OHG was estimated as a parameter, and then we multiplied it by a certain factor, and this was also estimated parameter to calculate conversion of 4OH to 4OHG as a fraction of the previously estimated clearance. Now, for renal clearance, the equation is shown here again, the allometric component and the maturation component, and we have direct excretion of midazolam, and clearances for hydroxy metabolites or glucuronide metabolites were estimated as basically a factor multiplied by the clearance of midazolam itself. Covariates that we had, again, we had many things, but since we didn't have too much data, very few patients, we looked specifically most extensively at hypothermia effect on clearance, and we tried to work with this as a categorical covariate, which didn't work well, but it was incorporated in the final model as a continuous covariate. So we had basically patient temperature normalized by 37 degrees, and we estimated a separate parameter for each type of clearance, so effect of hypothermia on on CYP-mediated clearance, on glucuronidation, and on renal clearance. We also looked at some of the potential effect of ECMO as categorical covariate on volumes and on clearance. And at the end, we also look at serum creatinine and renal clearance. Very briefly, the model was able to capture the data relatively well. We did have a lot of variability in some unexplained very high and low concentration, but again, we need to remember that all these populations were critically ill, and there potentially were several factors that we could not take into consideration. So we potentially need more subjects to improve the model. But what you can see here is individual predictions versus observed. The first row is for plasma data, the second row is for urine data, and then you have respective midazolam is on the right, and then here we have 1-hydroxy, 1-hydroxy glucuronide, 4-hydroxy metabolite, and 4-hydroxy glucuronide. So again, the first row is for plasma data, and the second row is for urine data. These were the estimated parameters, and the most interesting finding, really unexpected, was that when we determined these covariates, what we found out that instead of what was expected, that hypothermia will slow down the clearance, for us, what the model estimated was that the clearance was actually increased in all three different pathways in patients with hypothermia. Now, since each and every patient were very different, they received different dosing schedules, they were different ages, it was very hard to visualize all these effects of hypothermia on various metabolites and midazolam itself. So what we've done, we performed some simulation to really look at the effects of temperature, and we run it at three different scenarios. Either scenario one, when the patient is normothermic for the duration of the simulation. The second scenario, that the patient starts as a normothermic for a few hours, and then the temperature is decreased by half a degree every hour until 32 degrees Celsius reached. This hypothermic state is maintained for a certain period of time, and then the patient is rewarmed. The third temperature scenario was the patient is hypothermic for the duration of the simulation. And these are the results of the simulation. So normothermic status shown as a blue line, this changing temperature scenario shown by orange line, and the gray line shows hypothermia for the duration of the study. And we have here the midazolam concentration in the plasma, this left top. So the first row is plasma concentration, low row is amounts in the urine, and we have midazolam on the left, and then 1-hydroxy, 4-hydroxy, and then respective glucuronides. And we can see that the effect of hypothermia can be different on different chemical entities. We're moving on, we also performed this type of simulation at different scenarios. So this is a scenario 1, 2, and 3 to look at effect of different age. So in this case, the blue line, we're only looking on this slide on midazolam concentration. The blue line represents very young patients, so basically 2 or 3 days of age. And then the yellow line represents results for older patient, where it's 18 years old. And then the other two represents a median patient, which almost overlays with age for TM, which is about one year old. so we can see the effect of different temperature scenarios at different ages. And what was interesting that the patient started relatively high concentrations, and then this concentration would drop as a patient matures to the age of maybe 1 or 2 years old, and then the concentration will go up in an older patient. So we have two different factors pulling in different directions in this case. We have the effect of enzyme maturation and the body weight, increase in body weight. So what is shown on this slide, we have two different simulation scenarios where on the left-hand side, we're looking at a young patient, and we perform hypothetical simulation where the maturation of elimination pathway is just changing from 0.3 on the blue line of the adult value, 0.9 of the adult value here on the yellow line. So as expected, as clearance pathways mature, the concentration will decrease, right? Here we have patient of the same weight. This is, again, very hypothetical. On the right-hand side, I have fully mature pathways, but changing, again, hypothetically, the body weight of the patient from 10 kilograms to 50 kilograms. And we see that, again, as the body weight increases, concentration increases, and that relates to a higher dose, but also an allometric exponent for the clearance. So this is very briefly the results of our study. One more slide that I wanted to show and share here was interesting thing, that handling of the urine data. And one of the advantages of using Phoenix in this case that it doesn't really requires modification of the data set itself, because handling of the data is handled through the code. And I have a couple of examples shown here. Okay, and thank you for listening, and I'm open to answer any questions you might have.
Okay, so we do have a couple of questions. The first one is, what was the major challenge in analyzing this data?
Right, thank you, Don. So there are quite a few challenges, but I think the major challenge was too many factors that were in play and relatively sparse data. And again, sparse data, I'm not necessarily saying that patients didn't have enough time points. We basically didn't have enough patients in the study. Some of them had profiles that were quite extensive, maybe 20 or 30 points measured over time. But since there are so many factors in play here, hypothermia, ECMO, various ages, various sexes, I think this was one of the major challenges here.
Okay, thank you for that. And the next question is, have you tried to include peripheral distribution compartments for metabolics?
Right. This is always a very interesting question, how you identify some of these parameters, and identifiability of these volumes for metabolites is often a major thing. And again, since we did not have enough data in the data set, we had to limit the number of factors that we can actually specifically look at. And what we did here, we went with a previously described approach where the volume for metabolites was described as a function of the volume for midazolam itself, and we did not explore having more volume compartments for each of the specific metabolites. To really estimate those volumes, we would need data through injection of metabolite itself. So for example, injection of 1OH metabolite, and then following the pharmacokinetic of that metabolite following such an injection. In this case, we potentially will be able to resolve the number of compartments and volumes for that specific metabolite.
Wonderful, thank you. It looks like we are running out of time. And so thank you, Dr. Kagan, for your presentation. And we thank you, listeners, for joining us today. Dr. Thomas Henthorn from the University of Colorado School of Medicine will be presenting our next COE Express webinar on Tuesday, October the 4th. He will be speaking about lidocaine clearance during liver resection for a living donor transplant. We hope that you will join us then. Thanks again.